There we go. Okay, and this is just a reminder to our speakers, due to privacy law, this meeting is being recorded. All right, so here we are with using biologicals, best practices, and the latest research to grow cannabis. First and foremost, I want to let you know that Marrone Bio Innovations is a publicly traded company, and we are required to present this information to you at the beginning of our webinar. The information that we share in this webinar is purely for educational purposes to understand how our products work and what they do for you. It is not meant to help you understand whether or not to invest in Marrone Bio. So, of course, if you're interested in investing in Marrone Bio, we encourage you to do your own due diligence and make those decisions independent of this presentation. Now I'd like to go ahead and introduce our speakers. Our first speaker is my colleague, Nic Steeneken. He's the Northern California Territory Sales Manager for Marrone Bio Innovations. He was raised in Temecula, California, and has always had an interest in agriculture, animals, and nature. His passion for agriculture led him to Cal Poly Sonoma, where Nic graduated in 2018 with a plant science degree and a minor in IPM. Upon graduating, Nic worked for a large commercial cannabis greenhouse facility, and while working there, he saw a need for more professionalism and education within the industry, leading him to earn his PCA and QAL licenses. In December of 2019, Nic met Marrone Bio's Matt Breck at a key cannabis conference, and that started his journey to joining Marrone Bio first as an intern and then soon being promoted to Northern California Territory Sales Manager. So Nic is a very busy and active professional throughout the crop protection industry. He serves on the CAPCA board. He has often serving as an expert to our hemp and cannabis growers, and even spent some time out in the Midwest working on our new product for row crops called Pacesetter. So Nic has a wealth of very practical information for cannabis growers. He understands how indoor grow facilities work, and he's a great resource. So near the end of the presentation, we'll be sharing everybody's contact information. Highly recommend you make sure Nic's in your Rolodex. Our next speaker is our guest speaker, Stanton Gill. Thank you so much for being on, Stanton. He is a principal agent with the Central Maryland Research and Education Center with the University of Maryland. And he specializes in integrated pest management for greenhouses, landscapes, and nurseries, as well as cut flower operations. He's been conducting field trials to evaluate alternative pest control and low-risk pesticides for controlling nursery, landscape, cut flower, and greenhouse pests. He's also professor in the landscape technology program at Montgomery College, teaching pest management, greenhouse management, and nursery management. He's published in several books, including Pests and Diseases of Herbaceous Perennials, as well as, I'm getting caught up in all of the, as well as several others. I apologize. So yeah. Stanton is going to be on to give an overview of how to best control soil-borne insects. All right. And then my last colleague is Dr. Matthew Brecht, and he is our key account manager for Cultivated Garden. He resides up in the Pacific Northwest. And I apologize, Matt, in the busyness of the webinar prep, your bio has gone missing. So Matt is, I should have it memorized at this point, right? Matt is a owner of a cannabis operation. I believe it's Dutch Valley Farms. And he has, I think I found it and it just doesn't have your name on it. Here we go. He has a Bachelor of Science in Biology from the University of North Carolina, Wilmington, master's degree in plant pathology from the University of Florida, as well as receiving his doctorate in plant pathology from University of Florida. So Matt has worked for Syngenta and owns Dutch Valley Farms, a tier 1 indoor cannabis grow operation. And as I said, he resides in the Pacific Northwest, where he does a lot of his research out of his own indoor grow facility right on his home location. So Matt has a lot of experience with cannabis, obviously an expertise in plant pathology, and a good resource for you in your indoor grow operations. with that, I'm just going to quickly go over the agenda, a little bit of company overview, and then I'm going to hand it over to my colleague, Nic, who's going to get started. So Marrone Bio Innovations, we're going to first cover a little bit about MBI, and then we're going to go into some diseases and how to best control those diseases. Then we'll switch over to Stanton, who will go into root insect trial data, and then we'll finish up with best practices for managing insects. Okay. So first off, Marrone Bio, for those of you that aren't familiar with the company, it was founded in 2006 by Dr. Pamela Marrone. We are listed on the NASDAQ, and that started in 2013. We just relocated our headquarters to Raleigh, North Carolina, and we actually have facilities around the globe. So we have our subsidiary, ProFarm Technologies, based in Finland, and we have manufacturing in both the United States and Bangor, Michigan, as well as Europe. We have a nationwide sales and technical support, which we'll show you near the end, and we have global distribution. One of the things we're most proud of is our over 450 patents and 18,000 microorganisms in our research bank. Our current CEO is Kevin Elash. We have a broad portfolio of farmer-focused solutions, over 18 products in total. I'll go ahead and minimize this so you can see it. So our products range from crop protection to crop health to crop nutrition. Most of the products we'll be talking about today are in the crop protection section. For those of you that may have other endeavors as well as in addition to indoor grow operations, you may be interested in some of our crop health and crop nutrition products, which are specifically meant for outdoor row crops. We'll go into some of these products in more detail throughout the webinar. So now, actually, I apologize. I'm not handing it over to my colleague, Nic. I'm handing it over to my colleague, Matthew. So Matt is going to go over the latest powdery mildew, Fusarium, Botrytis research, and product deficiencies. And I forgot to mention at the beginning of the webinar, we were planning on having Dr. Punja on with us today. And unfortunately, he had a very last minute personal emergency this past weekend, which required him to attend to other things and not be available today. So he's in our thoughts and we're in close contact with him. He's doing well. However, just wasn't able to be on today live. So what we're going to do is try to do a follow-up webinar in a couple of months and have Dr. Punja on so that he can elaborate on his research that he's conducting. For now, Dr. Matthew Brecht is going to review a few things.
Yes, hi, and welcome to the webinar today. And unfortunately, yes, Samira is not able to make it. And what we're going to do instead is I'm going to show you some of the stuff that he has done over the years in cannabis for diseases. And here's a great slide that comes from one of his journal articles called Emerging Diseases of Cannabis Sativa and Sustainable Management. And this is a great read. I highly, I think you guys should definitely read it. But if you can want to start at the top and go down, he basically lists all these different diseases, Alternaria, hops latent viroid, leaf curl, stem canker, Fusarium, and so forth. They seem like there are a lot, but when it comes to an indoor facility growing, there's really only 3 or 4 maybe. And then in the outdoor, you sort of have them all in some respect to what you see there in some degree. And it also depends on where you are in the United States and around the world. Next slide, please. So this is interesting figure that he did in pest management science for the emerging diseases of cannabis. And what this shows is basically people are always like, hey, where does this botrytis come from? Where does powdery mildew come from? How did I get this Fusarium? What's going on here? And so this shows you that if you're growing outdoors, specifically either cannabis or hemp, these species of botrytis and Alternaria and Fusarium, powdery mildew, and sclerotinia come from different crops that we grow all over the United States and other countries. And you can see right here that, let's say if you're here in the Pacific Northwest, we have lots of blueberry and we have a lot of botrytis floating around. So just be aware of that. This is where the inoculum comes from. This is where your spores and diseases and things come from. So, next slide, please. Here is a figure that he did in Greenhouse Canada. This is a great read. Diseases affecting cannabis in greenhouses. And this figure really shows a lot of sort of the greenhouse diseases that we see on a day-to-day basis. If you go to the top left, here we have a Ipm infection, and you can see this sort of drooping. It's like wilting, very common for root rot disease. Okay, and the 2B is get more sort of like stunting and wilting. This is like sort of the initial stages of it. Trying to figure out why. It's getting enough water, but for whatever reason, still going down. That usually means, okay, something's going on with the roots. And then if 2C, this is kind of what you looks like, okay? And then if you look at 2D, which is the slide place right there, the slide with the auger plate, it's got a bunch of mycelium on it, and so you can basically pull out that fungal organism, plate it out, and yes, show that it is that fungus. And then, of course, if you want to make sure that it actually is the disease, you can actually put that back onto the plant and complete the cycle to make sure that it is the disease that it is. But we know pretty well that these diseases, Botrytis, Powdery Mildew, and Fusarium, are the major ones that we deal with in the indoor setting. And our products work really well with that. So I'm going to hand it over now to Nic.
All right, everyone. Thank you. So I'm Nic Steeneken. I'm the NorCal and Nevada rep for Marrone Bio. If you guys want to follow me, definitely add me on LinkedIn. I think that's the best way. I really enjoy being able to connect with you guys and see what you're doing and what you post as well. So I think it's a two-way street, and I really like that. Gonna get that out of the way. All right. Do I have control, Angela, or do you still have it? Let's see.
You should have control, yes.
Okay, sweet. There we go. You guys can see my cursor here. Let's see if I can turn the little laser pointer on. Oh, well. Laser pointer. Here we go. All right. So the plant disease triangle is a pretty fundamental basics for plant pathology. Any pathology class, it's probably the first thing you learn. So it's really broken down into 3 parts. You have a susceptible host. So in this case, it's the cannabis plant. Different strains, different varieties will have different susceptibilities to pathogens. So that kind of influences how big the circle is here. So basically, when you get all 3 different things, you have a favorable environment. Our greenhouses, our grow houses are perfect conditions for a lot of pathogens. And then you have to have the pathogen present. Essentially, all 3 of those factors have to be there. They all have to be lined up at the same time, and then you get a disease outbreak or a disease incidence. As time went on, people started turning this into a pyramid or a tetrahedron, whatever you call this thing here. And you add time on there and the human interaction as well. Let's see here, next slide. So moving even forward further, Dr. Surinder Dara out of the UC system here in California, I think he's actually moving up to the Pacific Northwest, kind of updated this into more of a holistic approach here. So he really broke it down into you have your pest management, you have your knowledge and your resources, your organizational planning, and then your communication. And really there's a lot of factors that go into a good IPM program. It has to be economically viable, environmentally safe, and socially acceptable. Your sellers have to be okay with the products, the producers have to be okay with the products, the consumers have to be okay with the products. There's so many factors that go in here. This figure on the left is actually more talking about plant disease sensing, but I just think it's a really good thing to bring up. You have your environments, your plants, your pathogens, and your management. They all interact in different ways. So I just kind of put this here as a little thinking point for you if you want to look up that. I think it's a really interesting article it came from. That was a UCANR article. And let's see here. All right. So really a good IPM program comes down to scouting, and scouting some more. Personally, I believe everyone in the facility should be trained, whether you're a processor, whether you're in there trimming, pruning plants, watering plants, you should always be paying attention to the health of those plants and making sure that they are in the optimal conditions. So I really think you should train everyone and foster an environment where if an employee sees something, they need to feel comfortable bringing it up and say, hey, I think I might have seen this. Hey, I might have seen that. It's a lot better to have good open communication and have your employees understand that, their observations are valued. So always look for signs and symptoms. So a sign of a disease is a physical manifestation of, let's say, mycelium or the fungal body, and then the symptom is something like a wilting plant. So there's a little distinction there with the terminology. Definitely think you should look into that if this is an industry you're interested in. So I personally think everyone in the facility should understand how to read your environmental scent points. So I'm talking about your link 4s, your different controller systems. Even if they're not necessarily authorized to touch the controllers and start changing things around, some of those systems do get complicated. You don't want everyone screwing around with it. Everyone should know how to look at it and understand generally what the parameters should be. I'm a really big fan of doing end of the day walkthrough for your facilities, making sure that everything is tip top, buttoned up for the day. You're not going to have any issues, no temperature swings, this exhaust fan got set to auto or to on completely, and then you freeze it out the greenhouse at night. Just really have everyone walk through and make sure. It's a really good way to close out the day, in my opinion. If your system allows it, you should definitely set up for remote alerts, and that'll just alert you to problems before they become a huge problem. See here, we'll go to the next slide. All right, so I'm going to talk about Regalia CG first. So Regalia is a broad spectrum product. It's acting on the plant's natural immune system. So it's an ISR SAR response, systemic acquired resistance and induced systemic resistance is what that stands for. So it's basically turning on different pathways in the plant. I'll touch on that in a little bit. The active ingredient is Reinutria saccharolensis, which is the giant knotweed plant. So it's actually a 5% solution that we extract out, and then that has the effect. If you're talking like big acreage, you're looking at 1 to 4 quarts an acre as far as rates go. For most applications, 30 mils per gallon per 1,000 square feet is really the sweet spot. That 30 mils per gallon being the point I would take home in most situations. It's a preventative mode of action, so it's ISR SAR response. A FRAC code is the Fungicide Resistance Action Committee. So essentially what that is, it's a nonprofit organization that kind of classifies all fungicides. So it's classified as a P5, which is a preventative fungicide with a biological mode of action, if I'm not mistaken. It's compatible with most crop protection products, copper products, sulfur products, parasitic acid, which is like jet ag or oxidate, oils, horticultural oils. You have like stylet oils, plays really well with stylet oil. It's a tank mix I really like. You can do foliar applications. If you planted your plants too close together outdoors and you have the right licenses to do it, you can do aerial applications. You can chemigate with Regalia, run it through the drip system. It's a pretty flexible product. I typically would say apply it where you need the control. So if you're looking at foliar control, do foliars. If you're looking at root control, do soil applications. Remember to always follow the label. Label is the law with all pesticide products. So talking about the mode of action here on the left, you see this nice little cell diagram here. So we have our pathogen invading it here. We have the first mode of action is really the ISR SAR response. So the plant cells are producing phenols, phytoalexins, PR proteins, all these different antimicrobial compounds that kind of act as a defense system. So you could think those the soldiers standing guard ready to attack if you break into the border. And then the cell wall is affected as well. So it signals the plant to lay down more lignin in the cell wall, making it more resistant, harder for that pathogen to send its penetration peg through and actually infect that cell. So think of that as like a strong border wall. So if that border wall is broken down and the pathogen gets its way in, then you have your army, your ISR SAR response, standing guard inside. The third mode of action is really kind of a benefit and kind of unique to Regalia. Not a lot of other products on the market have this, and that is that it helps promote plant growth. So you typically see more chlorophyll content, higher chlorophyll levels when you treat with Regalia. And what that does is more chlorophyll leads to more photosynthesis capability. More photosynthesis, you have more sugars, more energy for the plant, better growth, faster growth, healthier growth. Sometimes you see a greening effect as well. I've actually witnessed that in corn really well from the air. So Regalia is 1 to 4 quarts an acre, 30 mils per gallon. You definitely want to use it preventatively. It's not going to put out huge fires when you have them, but it will help prevent them pretty well. You don't need to add any adjuvants to Regalia. It does have a built-in surfactant in the formulation. It is rain fast in 1 hour. So that means, let's say you have a rainstorm coming and you want to get some Regalia on, limit the pathogen potential there. So if you get it on 1 hour before it starts raining, it'll absorb into the plant and you will be good. As far as washing off goes and things like that. And once again, it's tank mix compatible with most common chemistries. The takeaway I like to have about Regalia is if you're going to mix it, try to keep it under 1% solution volume to volume. So that's like 1 gallon of Regalia in 100 gallons of water. As long as you stay under that rate, you shouldn't experience any phytotoxicity. It is kind of a dark formulation. So if you're in, let's say, week 3 or after a flower, you may notice a little bit of pistil staining. Ways to avoid that are use lower rates, spray at night, not in direct light, things like that. It's OMRI registered, MRL exempt, 0-day post-harvest, minimal PPE, 4-hour REI, and you can definitely use in your organic program as long as conventionals, but pretty much all cannabis is organic, or at least it should be. Let's see here, next slide. So this is a picture I really am a big fan of. So the left here is strawberry plants that were treated with Regalia. The right picture was treated with actinovate and serenade. So it just shows that not all biologicals are equal, and you can really see the plant health response here. Let's see, the next slide I believe is from the same thing, but it's just showing the roots, and I think Matt might have some similar pictures coming up. So the left has been treated with Regalia and the right has not. And you can just see the increased in root growth, that nice flush of white growth there. Lots of fine feeder roots, really happy plants. I definitely encourage you, if you haven't done it, to take 2 1-gallon pots or small pots, take some clones that have pretty equal root systems. Plant one in, a 1 gallon that has, and then water that with Regalia after you transplant it, and then follow it up with some other applications as well. And you can see this for yourself in front of your own face, and it's pretty awesome. So here we're looking at tomato plants. Just this is a picture we had around, so I included it for you. And you can see the untreated control versus the 0.5% volume to volume. So that's like 0.5 gallon of Regalia in a 100-gallon solution. And you can see the increased root mass down there, more foliage, nice dark green coloration. They look like happy, healthy plants, if you ask me. Increased foliage, larger root system. And then this is just kind of a graph showing the chlorophyll levels. I know Matt has one that has some pictures as well, so I'm not going to get into it, but the important part is the Regalia has more chlorophyll. And I believe, okay, now we're on to Astargus. So Astargus is kind of a newer biofungicide. It's been around with Marrone 3 or 4 years now. Our label just recently expanded, so it added cannabis and a lot of other crops on there. It's Bacillus nakamurae. It was previously classified as Bacillus amylcaceus. However, as our genetic screening got better, we actually realized it was a Bacillus nakamurae, and it's been reclassified. The jug currently does still say Bacillus amylcaceus. Things go all pesticide products are EPA regulated, and they aren't the quickest at updating things. So expect that to change maybe in the next year or two, maybe sooner. So you're looking at 1 to 4 quarts per acre, pretty similar use rates to Regalia. And the ideal for cannabis growers is, everyone's milliliters per gallon. So 30 milliliters in 1 gallon of solution. That should treat about 1,000 square feet. It's preventative. It's really good at botrytis control. That's where Astargus really shines. Can be used for soil-borne diseases as well. So what makes Astargus unique compared to a lot of other products on the market is that during the fermentation process, when we're manufacturing it, we take this bacillus and we grow it out in a fermenter. And during that process, it produces lipopeptides. these lipopeptides actually will inhibit spore tube germination. So it has multiple modes of action. I'll talk more about that in a minute. But those lipopeptides are what's really important. It does trigger some SAR activity, so it kind of has some similar modes of action to Regalia. The frat code is BMO2, which is a biological mode of action with multiple modes of action. So the lipopeptides are really doing the heavy lifting in the formulation. They're the magic sauce in the jug there. It has living spores in there as well, which will colonize and help exclude pathogens. And then you also have some of that Isr SAR response. So when you apply it, let's say in week 2 of flower, you're worried about getting botrytis. So if you apply it to the colas that are forming, it will get in there, it'll colonize those colas. That's a nice, warm, humid environment. So you get in there with a beneficial species. It also has those lipopeptides, both produced from the bacteria and then in this formulation already, and those will inhibit spore tube germination. So you're looking at 1 to 4 quarts per acre. 30 mils per gallon is really the sweet spot for this type indoor application. That's 30 mils per gallon per 1,000 square feet. You typically want to do a reapplication 7 to 10 day intervals, especially with cannabis. It's a high value crop. It's worth it. For cannabis, you're starting applications in week 2 to 3 of flower for bud rot prevention. You could also use it at transplant time for Fusarium control in your transplant water, things like that. You can use it in rotation with Regalia if you want. Let's say you're using Regalia, but for whatever reason, maybe your environmentals aren't on point. You're still getting a little bit of powdery mildew. I've seen really good success adding some Astargus in that rotation every 2 to 3 applications. Let's see here. So it's compatible with most other products on the market. I will note that in California, we can use Astargus on cannabis. We're not looking at this type of bacteria, but some states have different testing requirements. So I would always make sure that you're in a state where Astargus will be okay and won't negatively affect your testing requirements. Some states have different ones where any bacteria on the plant is bad, which is kind of unrealistic if you ask me. They're living plants. They're going to have microbes on them. Unless you're, that's why if you've ever been into a dispensary in Las Vegas and you see some of the weed is dry and kind of brown and looks like it's old, well, that's because it had to go through remediation processes where it's been essentially microwaved and treated with radiation. So depending on where you are, just make sure your laws allow it. You can do foliar applications. You can run it through the drip. You can hand water it in. It's pretty flexible as far as what you're allowed to do. So this is just showing the spore germination here in a petri dish. So someone swabbed it on a petri dish and it grew in there, just showing that the live spores are viable. It mixes really well with water. It typically doesn't smell very bad. Some other bacteria products can get a really strong odor. Astargus typically doesn't have that. Occasionally you could have that, but typically not. So this is looking at Astargus's effect on Phytophthora and it's inhibiting the mycelium growth. So this is a 0.5% solution, that's a 1% solution, that's a 2% solution, and a 5% solution. So the untreated up here had pretty strong growth. The positive control, which is a conventional fungicide, controlled it completely. The 5% solution slowed the growth, but didn't completely prevent it. The 1% solution, as you can see here, it was pretty equal as far as to the positive control, so that's 100% effective. Going forward, you see there is no rate response. Once you get effective control, it is effective control. So when to apply Astargus? This picture on the left right here is really the ideal stage. Your buds are starting to form, but you can still get in there, get good coverage. This picture on the right here, it's a little bit too late. Those buds are kind of chunking up, getting denser, harder to get that material in there. And in this case, he should have really applied some either Venerate or BT as well, and some Astargus about 2 weeks prior to this. You can see a caterpillar got in there, killed the top of that cola, the bud rot starting right there. And back to you, Matt.
Wonderful. Thank you. Thank you, Nick. Wonderful job. Still got a little pointer. Look at you, got a little pointer. That seems pretty cool. I have to figure that out sometime. Wait, you might. Oh, I got the pointer now? Okay, all right, now this is weird. Well, so I'll be presenting a bunch of this data that we have for, or that's coming from some of the research that we did on cannabis with different research facilities. This slide right here is the effect of Regalia CG on the chlorophyll content of the cannabis plant, which is interesting. So, you know, we noticed, and growers have also told us that when you go ahead and spray, you know, with Regalia, in a little bit, maybe a couple days, you see this sort of green up happen with the spray. And, you know, some people will just say, hey, you know, maybe it's fertilizer or something. So we wanted to see, you know, what this was. And so Dr. Punjas here says that yes, he is actually seeing some chlorophyll differences. Now there's two chlorophyll in the plant. There's chlorophyll A and chlorophyll B. And chlorophyll B is not changed, but it looks like chlorophyll A is. And so with adding Regalia, you increase the amount of chlorophyll A, therefore giving them more of a green look to it. More chlorophyll means more energy, so hopefully a happier, healthier plant. So here's powdery mildew over time. This is the AUDPC curve, or area under the disease progress curve. And so if any of the researchers out there or people who've seen these things before, you know, these are my favorite graphs because this really shows disease over time throughout the study. And it really breaks out, I believe, products for their true sense of how they're working. And there's a lot of research that he did, Zamir Punja here, Dr. Punja, that for both powdery mildew, botrytis, fusarium, and I'm just putting out just a little bit of some of what he does. But if you want to see more of his research that he's done on all the products as well, in different areas, I definitely welcome you to visit some of his journal articles and read them. And so this slide right here really shows how well Millstop and Regalia controlled powdery mildew over time. And so if you see on the left, in the top left there, you have the disease rating. So that's a percent. So you go, you know, 0 to say the 30% disease. And the blue obviously is the control. So it's everything sort of starts the same. And then over time, the control should get worse, right? So the disease should come in and start progressing and the plant should go worse and worse. And we see that, okay? But what happens is that if you've used the Regalia or the Millstop, you can instantly reduce that disease over time pretty quickly to basically 0 to, you know, maybe 2%. All right. And so these are very effective products right now. And in Canada, you can only really have very few products. And these are the two big products that they're using in Canada right now. In Canada, it's called Regalia Max, so people just know for what it is. So here's Regalia efficacy on powdery mildew and Botrytis Bud Rot on cannabis. We did this in Ontario, Canada, at the Farm Forest Research Center in 2017. So on the left, you have untreated controls, okay? And what we're looking at is the percent incidence of powdery mildew and bud rot. Okay, so the darker brown is the powdery mildew incidence. The lighter brown is the bud rot incidence. And basically that shows you, you know, what they got over that treatment time period. And you can see right here that if you went out with the low rate, that you can not only reduce your powdery mildew, but also reduce your bud rot, and basically half, okay, without using anything else. And as well, if you go up in the amount you're using, let's say Regalia 2 course per acre, or this would be the excuse me, the 2, 3. This would be the 45 milliliter per gallon rate, okay, of Regalia CG. And so that we're looking at now here is so you get even more reduction in powdery mildew and more control of your bud rot. And if you go even the higher rate, you get even more as well. You know, typically what we say is if you use it, you should use Regalia CG at the low rates, start early, and keep going every 7 to 10 days. So here's Regalia increases marketable flower at harvest. So obviously, if you have healthier plants, bigger plants, and you're not losing that flower to bud rot, okay, then you've got more harvestable cannabis. And this clearly shows how over the time, you know, how destructive that bud rot was to if you don't do anything. And if you basically just treated it with a very low rate, you're increasing it, you know, significant pounds. That's significant, grams, grams in weight. Okay. So what we noticed that the higher rates of Regalia did increase the bud rot efficacy or control of bud rot, let's just say that. And we're going to go out. We're also going to talk about Astargus and how I like that as well as for bud rot. So we go to planting the Regalia, and here's some of the effects on those hemp transplants. Again, you see it's just a healthier root system, better uptake, maybe a little more chlorophyll. These are really cold, you know, tough times in the May of the southern Oregon that we have here in the high desert. So, you know, understand that even under tough conditions, these plants are doing a little bit better than the ones that are not treated with Regalia. Okay, and Nic has this slide as well. Regalia, you know, affects the roots right here at the growth, right at the transplants. And here's some stock that you see new feeder roots really coming on stronger because of the Regalia CG. This was an interesting, you know, we had a hemp farmer in Oregon, so Central Oregon. And he would just he sent us some pictures basically saying, yeah, the pictures or the whole area that we did, I think we did at least an acre of the hemp plants that had Regalia CG were at least like a foot wider in each direction, a foot taller in each direction. And they also had markedly less disease issues than the ones. And if you can look at the picture on the left, that plant in the bottom left there is riddled with Botrytis Bud Rot, and you can see the plants right behind it not having the bud rot at all. So the products they were using right here, both Regalia and just straight up Regalia, but using it quite often, worked really well. Okay, Stargas, we talked a little about the Stargas. How does it really work? Well, when we spray that Stargas on, that bacteria then colonizes the flower parts, the leaf parts, the stem parts, the calyx, everything, wherever it lands, and basically will exclude anything else around it, specifically Botrytis Bud Rot. So here you can see that on the top you see the Stargas, and it's fending off the Botrytis encroachment. Okay, so. I really like the Botrytis Bud Rot control with Stargas. It's amazing. So if you have really issues with Stargas, I got to tell you, you really should start looking at this. If you're not using Regalia or you are using Regalia and you still have Botrytis Bud Rot issues, especially if you're outdoors, definitely add the Stargas in for Botrytis Bud Rot control. And so here's just a graph right here. We see the untreated control on the left. So the little bar is early in the year, so it's like September, late September, and the yellow bar is mid October. Okay, and so you see the disease over time, and yes, obviously disease severity increases in the amount of bud rot that you see over time. Botrytis Stop is a product out there that has been around for a while now, and it has some decent efficacy, as you see. especially more of, I would say mid amount of disease severity. But if you really look at as far as control goes, Stargas really showed you a really good control. Anything, when you have 50% disease severity, I mean, you're talking pounds and pounds and pounds of loss, and that's thousands and thousands of dollars. And so even if you use a product and you're reducing it by half, you're saving so much money on loss. So there you go. All right, Astargus biofungicide reduces the Botrytis Bud Rot on the hemp, and here you can see the difference between the actual bud rot at the end of the trial. High pressure bud rot scenario. When you see an untreated control getting up as high as 66% disease severity, that's really high. Okay, and so Astargus applicable Astargus applications, you're bringing it down to 15%. Yeah, I think it's definitely well worth the amount of the little bit of money putting out to control your bud rot. Okay, here's another one. We're looking at disease of severity ratings. Okay, it's like a 0 to 5 scale, 5 being the plant's almost dead. Okay, 0, nothing's happening to it. Here we're looking at Botrytis Bud Rot versus some of the products that we have on at our disposal. Presta, Asperello, and then Stargas, and you can see that those products work decently here by a study by Dr. Zamir Punja on a cultivar called White Rhino and Afghani Kush. So that's a really nice study right there. So, but again, Stargas did a great job, 93% less disease than the water control. Fantastic results in this product. Here's another one. I don't go to many of them. But data and research matters when you're talking about efficacy of what are you going to talk about products. And here we have we're going up against non-treated control for botrytis. This is also in a greenhouse in Ontario. Percent infested buds. So 18% of the infested buds are the non-treated. And with Astargus, we're looking at somewhere only about 2, maybe 1.9%, 2.3% in that trial, up to maybe 3 to 5%, 3 to 4%. Okay, that's about it. And with those couple applications of Stargas, right at about say mid flower to mid to late flower of your grow. And we'll talk about a little how you put it out there a little later. But for right now, I'd like to turn it over to the one and only, the great Dr. Stanton Gill, to go over the root insect trial and data and pests. Dr. Stanton, are you there, sir?
We might still be waiting on Angela getting all the slides pulled up.
Okay.
So we're just going to go ahead. So let's take a moment, and any questions you have for us, we're going to try to run through them now. And if you have any comments about like cultivation practices you like, just let's share them in the chat. Let's get a little interaction going here. And I think maybe it looks like we're pulling it up, so maybe we don't have to do this. So Johnny asks, I can spray potassium bicarbonate on the flower to control botrytis, and I can reduce the production of the resins. So Johnny, are you saying that in your experience spraying millstop would reduce the production of the resin?
So I will say that, man, I wish someone from Canada was on. I know that millstop's label in Canada may not have during flowering. I think I remember that from someone, but it might be one of the reasons. I'm not really sure, but our products won't do that. I know that for sure. But potassium bicarbonate, which basically is millstop, may. I don't know if that's for sure.
And so Mohammed asks, is there any evidence about efficacy of Stargas on Fusarium bud rot? So to my knowledge, most of the bud rot that we've looked at is usually Botrytis bud rot, which we know it's very effective against. I know it has an activity against Fusarium in the soil, so preventing that infection getting into the plant should limit the amount of bud rot you're seeing caused by that. So I guess the answer would be yes, indirectly, but I don't know if we've actually
Yeah,
quantified
Nic.
the bud rot reduction. Sweet.
Yeah, so Nick, so so I think what you're talking about is or here, so you're talking about is the aerial Fusarium blight that we see. That's quite often, especially in the eastern half of the United States, where it's a little bit more humid. So we do see that. Does it work on Fusarium? We do have some activity on with Astargus on Fusarium. It's on the label. I don't know if it's the best thing ever we have. I think Regalia is a little bit better. But if you have serious issues with Fusarium issues, definitely that is a product that will help you with it.
And we have a 2EE recommendation, which is a California specific recommendation for Fusarium after transplant with Stargus. So you can check that out on our website. And I think it should be ready for Stanton.
Okay. All right. You want me to jump in?
Yes, please, Stanton. And I unfortunately can't give you control for
so
whatever, because you're not within. So I'll click through for you. Okay.
Okay, great. So the
right.
project, and by the way, my name's Stanton Gill. I work for the University of Maryland. I'm an extension specialist at the Central Maryland Research Center and a tenured faculty member with the University of Maryland, and also a professor at Montgomery College. Brian Kunkel is with the University of Delaware, and he's a fellow entomologist that worked on these projects with us over the last 2 years. And Tom Ivento is a statistician that is at Delaware University that was involved with the stat work. And then Suzanne Click is our technician here at the Central Maryland Research Center. So we move to the next one there. Next slide.
Okay, thanks.
There we go. All right.
There's just a delay.
So,
sorry.
Yeah, a little bit of delay on that. Okay, so the two insects we're looking at are root mealybug and root aphid. Both of these are things that have been overlooked by a lot of people in the industry because they're down in the root zone in the substrate and often go unnoticed. The plant may be showing effects on the top and people are focusing up there and not looking down in that root system. If you look at this aphid on the left, it's typical aphid shape. It's narrow in the front, a little wider in the back. Most of the aphids you're going to see on your crops and your greenhouses are going to, on the tip of the abdomen, have two structures sticking out of the back called cornicles. And they look like little tubes sticking up. Some people call them tailpipes. On the root aphid, they're very, very reduced. And if you look at this picture, you really can't see them on the shot. You'd have to zero in and magnify that around 20 to 30 times to be able to pick up small little bumps there that are remnants of cornicles. But like all aphids, they have a sucking mouth part. They jam it into the plant root and they're extracting juices. They're after the protein in there. And what they get is a lot of water and sugar. And that sugar, just like a diabetic would excrete sugar in their urine, basically out of their waste material comes a honeydew, comes out of the rear end. So you get a sticky material that comes out of that. Not as much on the root aphids as you'd see on the ones above ground, but anyhow. All right, so move on to the next one, please. On aphids, we have winged and non-wing forms. The wing forms are called alates, A-L-A-T-E-S. And an alate is usually for dissemination. If you look at this is that same insect. That's the adult, the wing stage, the late stage. And if you look at the wings, they're very, very large. They're 2 to 3 times the length of the body, and they look like they're almost inverted over the body when they hold them up over there. But these can be out in a weed area, a wet area, whatever. They will fly into your greenhouse. If you don't have micro screening on that greenhouse, you could have them show up in there. And once they get to the plant, they're going to begin to live birth. The vast majority of these rice root aphids are females. It's rather rare to find males. Most of the females reproduce parthenogenically. So once they're in that soil, they're going to start birthing out young little aphids, and then they'll move down into the roots. Those ones that are on the roots will not be a wing form. Flight is very expensive in the animal world. So if you fly, it means your body has to narrow up, get a little bit lighter. For best reproduction, they're going to stop having those wings and just reproduce themselves down in the soil. Next one, please. What you would pick up is if you had a plant in a container and you pull the plant out of there when the roots have fully engaged the substrate and everything. If you look on the edge of that pot, you'll see a white powder that you're seeing up on the right side of here. The aphids will have little micro and macro pores on their body that they excrete a wax from, and that's what you're picking up. And that's part camouflage and part defense for them. And if you look on the left, there's a little red circle there. And that's how it would look with your naked eye. If you have 20/20 vision, that's what you'd be seeing. They're very small, and you can see how these would have gone undetected by many growers. You know, on both those circles you can see there. Okay, next one, please. Here you can zero in a little more, and you can see where the arrows are pointing to aphids. So it's focusing you in. This is magnified. This is 12x magnification. You see the one in the center with the red arrow going down, or orange arrow. That's aiming toward an elate. All right, but the other ones are all non-wing forms on the plant. Okay, next, please. Again, so here you're looking at the root system and see the two arrows pointing in there. Now, what you need to do is train your eye to pick those things up. The coloration often blends in with that root zone. You can see the roots are pretty much the same color. Small little creatures. They're not overly active. Aphids, when they settle down in a plant on that root, they're going to put their stylet mouth part in there and pretty much stay put. So there's not a lot of moving activity that you're going to pick up out of there. But they're usually, we find them in most of the containers toward the edge of the pot. That's fortunate. In our studies, when we were digging through those, we found some in the center, but the vast majority are on the edge. A few are on the bottom of the pot. Okay, next one, please. And here you can see a typical clustering. What will happen is the female that reproduces, and this is usually parthenogenically, so she doesn't need to mate with a male. She just live bursts out. She doesn't lay any egg or anything, so everything is hatching inside of her body. And actually, if you took her body and put her underneath the scope, cleared that body, you'd see a series of young little nymphs inside of her. And then if you look inside them, there's another set in there. It's a little bit like the Russian nesting dolls that you see. But that female, the one in the center, the largest one, is the matron there, and she's birthed out these ones around there. They tend to stay in a cluster like this. So when you build up a population and you're seeing a cluster, that means you're probably in trouble. You have a very high population of those in on your crop. All right, next one, please. Now, the interesting thing is when we were doing our studies, we picked up aphid species in there. There's a thing called a Citronella ant that we find pretty much across the United States, and I suspect you find it out in California. Citronella ants are very common. They give off a lemon odor or citrusy smell when you handle them. But we found them in the container, the plants we were dealing with, actually tending aphids, much like we would have a cow out in the field or a goat or a sheep that we're milking. They're basically milking these aphids. So what they'll do is they will come up to them, they use their antenna, and they will antennate them. They will drum on the outside there to stimulate them to produce honeydew. All right, so that sugary material that comes out of the rear end is a benefit to those ants, and they'll carry it back as a food source for their colony. So ants are not your buddies in this case. We found them down through the soil, tending these aphids, and if a predator or parasite comes in there, they'll actually defend them, the aphids, against that predator. Yeah, interesting. All right, next one, please. Now, our studies realized we were working on ornamental crops, but a lot of these things, I know you guys are involved with the cannabis crop. Here in Maryland, I can only work with people who are doing field hemp. And we're not allowed to work with the medical marijuana industry because we're federally funded besides being the state. But go ahead on the next one, please. So the crops that we were working with, we were able to obtain root aphids. It's kind of interesting. We see them in sedum crops in our many parts of the United States. They're going to using sedums on green roofs now. And so the plants are being produced in large quantities, and they will see root aphids build up, and root mealybug, the next one I'll talk about. But we have many aquatic plant nurseries that are producing plants for using around things like the Chesapeake Bay here in Maryland. And we happened to find a nursery that had aquatic plants, juncus plants, and bulrush that were heavily loaded with the aphids. And we needed that aphid population to do the studies. We pulled the aphids out of there. You had to pull females out, and we sent them down to, and actually in entomology, we have different specialties. We have people that specialize in these called aphidologists. Susan Halliburtt is with the University of Florida, IFAS, and she's one of the leading aphidologists in the area. So we send samples down to her to get a confirmation that we are, which species of aphid we're dealing about on the root aphid. And it was the rice root aphid, which is fairly common across the United States. And you find it in aquatic environments. And it's called the rice root aphid because it's usually found on rice, but it does show up in these other crops, aquatic plants. It'll show up in your cannabis crops and seems to have adapted to them very nicely. Next one, please. So these are some of our trial blocks when we pull the plants out of there. In the nursery case, they were growing them as plugs, and their customers were noticing when they were shipping those out that the population was so large. What their customers really picked up on was that white wax that was showing up there. That becomes very noticeable. And maybe they didn't realize the aphid because they didn't know what they're looking for, but the growers know what to look for now and that. But it can build up quite heavy. They were getting stunning of the plant and dieback. Now, the interesting thing we found with rice root aphid, like a lot of the aphids, it likes the cool weather. And we tend to see those building up in the early spring. And here in Maryland, we'll see that in March, April, May. As we get into the later part of June and July, when it gets into the hot weather, we'll see a natural decline on the root aphids. And so you may see it in your greenhouse environment as it heats up and gets warmer and warmer. There may be less of a problem. Other times of the year, it may be a bigger deal for you. We do have cannabis growers out here in DC of Maryland that can grow the crop, and several of them under artificial lights, and they've been sending us samples up of these root aphids showing up in the plants. Seems when you grow in a container for any of the cannabis crops that they seem to be highly susceptible to these rice root aphids. Next one, please. Okay, so again, you need to look down in the root zone. Some of these, when you flip that over and you're looking at a root zone like this, it'd be very difficult to pick up where the aphids out of there. Okay, so again, you have to train your eye. The idea is you want to catch them when the populations are low. So spend a little time looking at the pictures. You know, you have my PowerPoint. You can blow those pictures up and take a look. You want to catch them early on. But if you look at that picture, you might begin to pick up an aphid in there now that you've seen some earlier ones. But you look at that mass of roots, you can see why a lot of people just breeze by this. When we just published our results of our studies in the Journal of Hort Science, and we had to review the literature. And most of the work that's been done was quite a length of time ago. No one's done anything currently. And I think because these insects are so difficult to deal with and also to get a good population to do a study on. Next one, please. Now, some of the materials that we were testing out, we're interested in looking at, we use integrated pest management out here in Maryland very heavily. And we have trained scouts that we send out to different greenhouse operations to do scouting. And what fits into those programs are things, there's several products from Syngenta that's put out, Mainspring and Aceleprene. These are diamide chemicals. Endeavor is a insect stylet blocker, which would work on things like aphids. It basically clogs up their stylets so they would starve. The Granvio and Venerate are materials that are from Marrone that they sent out for testing, and these are on the market, of course. The other ones were some numbered numeric alphabetic compounds they had us involved in the trials. And then we put it against a entomopathogenic fungi called Bavaria bassiana. And you can get that under a couple of different brands, but BioWorks sells that under Botanigard. And that's the brand name we use for this study. All right, next one, please. Now, and I had the rates on there for the different ones we used, if you guys want to refer back those later, because we only have a short time to go over all this. But basically, when we took pre-counts on all those plugs, which involving about 6 interns and using the scopes because they are so small, and to establish if we had population to begin with before we started any of the treatments. The controls were treated with water, and the other ones were treated with the different materials. Data was taken at 14 days and 28 days. Now, realize something. When we do studies on different chemicals, and whether it's root aphids or any of these things, generally we'll take data. Some of the things are as short as 7 days, but generally it's 14 days before we begin to see any activity for most really impact on things. You can sicken an insect, but before it dies, it usually takes a little bit of time. The older classes of chemistry, like the pyrethroids, you can expect them to die within a day or so, but a lot of these other things just aren't as quick. At 28 days, we really begin to see the impact for a lot of these. So you have to learn to be a little more patient with them, and we point that out to our growers. Okay, next one, please. There's some of the products from Marrone that we tested, and you go on to the next one there. And then we had some from BASF. We had things that were from BioWorks that were sent into us for testing. That was the Bavaria bassiana. Velsifer from BASF is a Bavaria bassiana, the animal-pathogenic fungi for insect control, and they mix it with an oil. So we evaluate that in there also with the trials. Okay, next one, please. All right, so the thing on the left is pre-counts, and it was not even by any means when we did the pre-counts on there. You could find on a population a heavy one, light on the other. It just was, but we did have a population on just about all the plants, and we selected those out. We had to go over close to 600 plants originally to pull out enough for the trial so we could have the replication in there for the study. And so we had an established population is what it gets down to. Listing the materials there, if you look at the WAT, that means weeks after treatment. If you go on to the next slide, please. Okay. And you can see, again, on the pre-treatments on the left, just showing a population in there. These are the different materials that we tested. If you look at the black thing on your right, the histogram, the bar that's sticking up there, and there's looks like a little T sitting over those, they're called error bars, okay? And that, if you're doing any good science project, you're going to have error bars on it. If you don't, I kind of question what person did with that. But anyhow, that shows the control. The population stayed up on the water treated control. What we found is most of these materials we were testing as a drench worked very well. The thing is, getting the population when it's small like that and putting them on early at 28 days, they were all fairly effective on it. Next one, please. Okay, the Bavaria bassiana was the interesting one, being an entomopathogenic fungi. We're interested whether that would work, and especially in something like common rush and bulrush, where they're growing it in a wet environment. It was perfect for that. It worked very, very well. It really reduced the population very nicely. But we saw several of the materials worked well. Some of the number numeric compounds from Marrone performed very, very well for us. The mainspring and the aceleprene from Syngenta were also very, very effective on there. And we've had growers use that as follow-up and got very report back very effective control. Next one, please. Now, it's interesting, when we were doing the study with the rice root aphid, we picked up a thing called Cryptolaemus montasuri, which is commonly called the Mealy Bug Destroyer. And we've used that in the past for things like mealy bugs that are above ground. Things like citrus mealy bug is common on Madeira mealy bug. It works pretty well on. But I hadn't seen that go down in the soil. I didn't realize it went down in the substrate, and I didn't see where it's been published in the literature. So we put that in our referee journal, the report we found those. We also found rove beetles, which are a predator, that were down in that substrate feeding on those aphids. So we do have some good guys in there. We also found some of the Stratolaelaps, which are mites that live in soil. They were in there around the aphids too. So there's quite a colony of good guys in there. So one of the things we need to look at for the screening in the future is these materials to see what impact it has on these good guys, because we want to preserve them as much as possible. All right, go on to the next one, please. Here, it looks like you're looking at a mealy bug. But the thing on the left, of course, is that root aphid. But the thing on the right is actually a Cryptolaemus larvae. They put wax out of their body, much like a mealy bug does. The way you could tell them apart as a grower, I tell people it's very easy. Flip it over, if it was a mealybug, they're going to have a sucking mouth part, just like the aphid would. So it'd have a stylet straw-like mouth part sticking down. If there's two mandibles out of there, that's a chewing mouth part. Well, then that's a predator. Okay, that's the going to be the Cryptolaemus, which is a type of ladybird beetle. But they were down in the soil. We went back to the grower and mentioned to them that we were picking these up and they were feeding on aphids in there. And they said, oh, well, last year we did a release. They had citrus mealybug in there and they released them for that. And we said, well, guess what? They're moving down in that substrate, going down into the ground. And so they went back and looked and they found there was quite a population in there and they were doing a very nice job of cleaning up these aphids. Okay, next one, please. So there's some biological control that have possibilities. We picked up things like crab spiders in the substrate also, and they were feeding on the root aphids also. Next one, please. And here you can see one of the spiders in the root zone right there. They were very active walking around. The aphids don't move much, but these guys were very active. The rove beetles were very quick on moving through the soil, and they were doing feeding too. But going back to those ants, if the ants are in the soil, they are going to defend those aphids and are going to fend off things like spiders and the Cryptolaemus. All right, so it's a little bit of war on that. Next one, please. So we got our own Russia Crimea thing going on with that. All right, so the other one we worked with is if you move on to the next slide, is the root mealybug. Okay. All right, so the big thing we were working with was originally when we started this trial wasn't cannabis, but it was really the sedums for green roofs. And say green roofs are very, very popular in a lot of the city areas where they're the flat roofs. They're trying to put plant material up there that can take the heat, deal with the water runoff, and have something that comes out of the drain that's clean. So the extremely popular plants, when they were originally installed, everybody said they were insect free. We know anything you plant in large numbers is going to get an insect problem. And these mealybugs, root mealybugs, showed up in there. Much like the root aphid, they're all below ground. They're going to be white in color because they're not getting any light on them. They don't have any coloration to their chitin. Okay, next one, please. Okay, so on green roofs, they say that's the big thing. You can see this is over at our University of Maryland where I work on one of the green roofs there with the sedum plantings in there. Lovely little plants. That's about it. All right, go on to the next one, please. Much like the root aphid, when you pull a plant out of the soil, you're going to see this white wax. And we have growers trying to identify the cause and say we have white wax, so therefore we have, and they'll proclaim they have root mealybug or they have root aphid. And we said, did you actually see the insect? No, we see the white wax though. Both of them do the same thing. Okay, you really got to look close on that to pick up that it's a mealybug, and I'll show you those a little bit closer. So the aphid has that distinct narrow front head part to it. It widens out in the back. And if you look with a hand lens and magnified, you see the reduced bump cornicles on the back, which you're not going to see with this root mealybug. Okay, next one, please. Now, interesting, root mealybug, where we usually see it start out is when people do plug production. And when the plugs, they tend to be on the very, very bottom of that. They don't seem to like the side as much as when you get to a bigger plant. So I know many of the cannabis crops are vegetatively produced and they're started in a greenhouse. When you get a plug, look and make sure you don't have root mealybug down the very bottom of that before you put it into the pot and introduce the problem into your greenhouse. Once you have it in there, it is going to spread between pot and pot. I mean, it will go out the bottom through those drain holes and they will walk across many of you using white plastic on the ground for those things. They'll walk across that. Okay, go on to the next one. You can see at this nursery, again, they were doing sedums, not doing the cannabis, but they have little plug trays that are sitting down on these raised kind of plastic devices to keep them up off the ground to help them drain. And then there's a weed barrier underneath there. And what we found is if you have infested plants, you'll have an epicenter where they will start, but they will go out the bottom of those the flats through the hole and walk between that like a little super highway, and they go between the plants and you'll move it right down through and infest just about all the plants. Same thing happened in with the cannabis crop when you're growing it in the containers. They'll come out the bottom of that, they'll zip across there and go in the next pot and infest that. All right, next one, please. There's not a wing stage with this insect like there is the elates that you see on the root aphid, but they certainly are very mobile unto themselves. We started projects on this in 2018, and then we expanded as we went along. Some of the early materials we used was Aria. Aria is very similar to the Endeavor that I showed you earlier from Syngenta. Aria is from Gowan Company. That's a stylet blocker. Again, it jams up their stylet. They basically starve to death. Altus is a new material from a Bear Company. It's close to a neonicotinoid, but it's not. It is a true systemic, and it's performed very well for trials we've done with whitefly and with other aphid species. So we threw that in a trial. The Acetylpine and Mainspring were in there, and then the Endeavor on that one in 2018. Next one, please. Okay, so the way we're applying these are in plug trays. So we would mix up the solution, and then we would make a drench tray. We took a stainless steel catch tray down below, and the material was poured through those to soak it down. On root mealybug, they're going to be on a small one like this. They tend to be on that bottom of that plug, as I mentioned earlier. As you get into a bigger plant, you're going to want to drench the sides of that pot where you get most of that material, and on the bottom, because that's where you're going to find the population in heaviest numbers. In the center core, you just don't see as much on those bigger ones. Next one, please. So this is, again, the pre-counts on your left, and then on the right, see that large black one is the untreated control. You can see several of these, the Aria, the Altus, the Acetylpine, all those work very well for us. The Mainspring on the one treatment, on the one rate, not quite as good, but the others just knock the heck out of it, work very, very well. Okay, next one, please. This is another chart on that. You can go on to the next one, please. In the follow-up trials in 2019, then we were asked by Marrone to include in the trial the Venerate, which we did use, and we used Bavaria bassiana, again, that entomopathogenic fungi mycotrol. We had Granvio in the trials also, with 2 different rates on that, and then of course our Mainspring, and then we tested against a entomopathogenic nematode called Steinernema carpocapsae, because that had been reported to work on root mealybug and some other studies by other people. So next one, please. So again, the pre-counts on the left, you can see we had again, varying population on there, but we definitely had an established population. On the post counts, again, that large black one, you look on the right, that would be the untreated control. It's way up there. You can see just about all these materials gave a fairly good level of control, some better than others, but still quite appreciable. Some of the better materials were the Mainspring, and the Acetylpine was probably some of the leading ones. The Venerate did fairly well out of that. The Steinernema carpocapsae actually surprisingly did fairly well on that one, which was interesting. Bavaria, a little more variable on that. Okay, next one, please. So our conclusion: root mealybug is difficult to study, and because it's below the ground, just like that root aphid, difficult to find population of those for a trial in there. We did 3 years of work on the root mealybug, and we did 1 year of work on the root aphid. All of this has been published in the Journal of Hort Science. If anybody would like a copy of that, it was just published in December of 2021. I have PDFs available. If you go on to the next slide there, please. You can get a hold of me. It's very easy with people at the university. It's usually their first initial and their last name. If their name is short on the last name, my name's nice and short. So it's sgill@umd for University of Maryland.edu. And if you'd like a copy of that, I'd be happy to send that along to you. And if you have questions, certainly you can send them along, and we'll follow up with you on those. And hopefully you don't bring root mealybug into the greenhouse is a big thing. The root aphid, not a whole lot you can do other than have screening on that greenhouse to keep it out. It's the best thing I can tell you. All right. Hope you got something out of it. Sorry, I didn't get to move this forward, but you did a wonderful job there on your end over there, moving them forth with the go forward type thing. All right.
Awesome. Thank you.
Question?
Yeah, thank you, Stanton. I'm going to ask my colleagues, Nick and Matthew, if you could look at the chat for me and see if there are any questions that we can pull up for Stanton while I go ahead and switch the deck. Stanton, it just seems like this webinar, it's been one thing after another preparing for this webinar. So thank you for your patience and understanding. We had a webinar yesterday. It went as smooth as pie. And now today, you know, hiccups. So appreciate everyone's understanding. Nick or Matt, do you see?
Yeah, I'd like to say,
Okay.
I'd like to say something. And I know that Stanton's got some great, fascinating research. It's just amazing, some stuff I actually didn't know some of the stuff. So it's a great refresher and some new stuff. But Stanton, I know you were talking about some of these insecticides that you guys researched. I just want to make sure everyone on this cannabis webinar understands that products like Acelepryn, Mainspring, Endeavor, and some others that he talked about are not allowed on cannabis.
Right.
And so every testing facility is testing for every one of those active ingredients that are not allowed, obviously. So the ones that are perfectly fine on cannabis for most states right now that have cannabis growing in them, for that matter, would be the Venerate and the Grandivo CG. The CG line is different than the CG line is different than the agricultural line, just to make sure that everyone is aware of that. There's special language on the labels for the CG line of products that allows their use in the country, let's say United States, or in Canada. Okay, so these labels are very specific, has very specific language. And then lastly, all CG products go through a second level of testing. We send our product down to SC Labs to do a full evaluation on heavy metals and other things. So that way, none of our products will none of our products will lead to a failure of your test. However, I will say this caveat is that some states are testing for biologicals on their flower, so that could be any bacteria or fungus they pick up. So if you're applying, a living fungus, let's say, Bavaria gaussiana or our product Astargus, you may pop hot for a biological. So just keep that aware. Okay.
Yeah, the one thing, and those are good points, and thanks for pointing out about the labels. The things like the animal pathogenic nematodes, the Steinernema carpocapsae, I believe that could be used on the crop. Is that correct?
That's correct.
Yeah. Okay, and any of the biologicals that, such as the Cryptolaemus, those certainly could be used there. Yep.
Yeah, and I think what's fascinating is your findings of some of these on root aphids or on root mealybugs. I'm actually astonished by that. Years ago, at one of the first bio controls west here I've been to, they said something about rogue beetles, but people weren't really sure about it. Now you said it as well.
I wanted to. Yeah, I did one point. Rove beetle we've used with greenhouse operations out here in Maryland for controlling things like fungus gnats, actually. And usually that's in a rooting area where they have it under mist, and it's worked very, very well.
I have two questions. One, root rockwool versus, let's say, if you're growing cannabis in cocoa core or peat moss perlite, what I understand is that some rockwool growers can almost grow through it because the root aphids don't necessarily get inside their rockwool. Can you talk about that at all?
No, I cannot because I haven't done trials with it. It did come up when we were doing the work on the root mealy bug. Some people had reported that if you grew up on a bench area with wire or screening, that the air underneath would dry that out, and they wouldn't migrate between the plants, or the survival was supposed to be reduced. So to that end, we set up a trial at the Delaware University greenhouses to evaluate that. And we could find no significant difference between the ones grown on the ground, where they had plenty of moisture to walk between things, and the ones up on the bench. So that drying out had no impact there. Yeah, but there's lots of neat studies like that need to be done. Sure.
Well, I have more questions, but I think we're going to turn it over now to Angela.
Okay.
I think she's got everything back up and running.
All right. Well, you guys have a good rest of the day.
Right. Thanks so much, Dan. Are you going to be around to take more questions, or are you going to be leaving?
No, well, you have to remember, on the East Coast here, it's getting rather late in the day. Originally, I was told it was 8:30 in the morning by someone, and then it turned out it was 11:30. I thought 11:30 East Coast, but remember, we're 3 hours behind, ahead of you guys, so it's a little late in the day for me to stay. So, yeah.
Okay. Well, I have one more question. I have one more question. I wanted to bring this up was, knowing our safeness on beneficial insects with Grandivo CG and Venerate CG. Are you planning on looking to see, the effects on these root beneficial insects as a fight off root aphids and fight off root mealybugs?
Well, it's funny you're asking that question, because when we put out for the journal article, of course, you go through that year process. Several reviewers put on there they want more studies on the impact of the beneficials. We just observed them in trials, but that wasn't the mainstay of the studies we did. And we said, yeah, that's for future work, but we had to obtain another population. The problem we've run into, we gave the information out to our growers and put it out there, so they're applying these materials and now getting great control. It kind of shot us in the foot on trying to get a population now. So we have to find another population to do that. Yeah.
Well, Stan, let's keep in touch, because I have other populations of these insects for you at any time. So.
Okay. All right. Excellent. You guys have a good rest of the day.
Thank you so much for your time.
Thanks, Stanton. All right, I'm going to hand it over to Nick. We just want to be mindful of time, everyone.
All right, so I'm going to start off again by saying thanks for all the people that have added me on LinkedIn. I had 5 people add me in the last 10 minutes, so that was kind of cool. So I'm going to answer 3 questions out of the chat here. MP'd is not our product. I know in California, I think it's on the approved list for cannabis, the list that's not a list, but that's not our product. So Astargus has kind of that broad label verbiage for cannabis use included in the master label for Astargus, so we didn't differentiate that and make a CG line, but Astargus just has all that built into it. Rove beetles typically don't mind the Venerator Grandivo. The products really need to be ingested to have activity. Yeah, I think that's all of them. All right, I'll carry on now. All right, so this picture here is actually in Natura Life Sciences in Sacramento. That's the founder of Marrone Bio right here, Pam Marrone. She got to join me on this tour of this very high-tech greenhouse facility. I put it there because I think it represents a lot of the aspects that are important as far as excluding pests and diseases. So something to think about, and please add to my list in the chat here if you have anything that comes up. I think there's a lot of things out there. This is kind of what was on the top of my head. So changing your clothes when you get to work, not bringing outside pathogens in. Definitely you want to be cognizant of like soil pathogens on your shoes, especially if you're growing in like raised beds and you're required to like walk in the bed. I know you see some of the people up in like, let's say Humboldt County going barefoot in their raised beds, because that way you're not bringing dirty shoes on there. Probably grounds you with the plants as well, but I don't think that'll happen in most indoor facilities. If you can filter the air, that's a great thing. Same as using drip screens on greenhouses. You definitely want to screen your intakes and your vents if possible, including ridge vents. That's something I often see overlooked, is the ridge vent as a entry point. When you're bringing in new plants, bringing in clones, bringing in new mother plants, new genetics, you definitely want to quarantine those plants, inspect them. Some people will even send them out for viral testing, things like that. If you know some areas of your facility are cleaner than others, you're definitely wanting to work from the clean zones into the dirty zones don't start your day in the zone that's full of aphids and powdery mildew, and then go work in your clone room afterwards. That wouldn't be a good idea. You want to do that the other way typically if possible. Or, change clothes, use coveralls, minimize that transfer. You definitely want to clean your pots and any materials, any cultivation materials, if you're going to reuse them. So what trick we used to do when I worked in the greenhouses was, it gets pretty hot in the summer in Southern California. So let's say we finished a harvest, cleaned out the pots in that room and weren't going to refill it right away and could fit this into our schedule. We love to just close it up, close all the vents, and just let it bake and get really hot. That'll help kind of reduce some of those pathogen pressures and insect pressures. I think it's just a good thing, as well as cleaning everything, sanitize it. If you're in an indoor environment or in a real fancy greenhouse, if you can have a positive pressure, that's great. That way you minimize, let's say, things getting sucked in when you open the door, things like that. That would go along with proper filtering as well. Keep your cultivation area free of dirt and clutter. If you have a bunch of stuff sitting in the front of your greenhouse, or if you have potting soil that fell into, let's say, the bottom of your light trap for your depth curtains, and you have some stuff there, that could definitely be a source. Pests could hide there, pathogens can hide there. You can't effectively sanitize something if you're not cleaning it first. You have to clean it, remove the dirt, and then you can sanitize things. If you're outdoors or even in a greenhouse, clearing the surrounding brush, just give yourself a little bit of a buffer zone. It's good for many reasons, but the main one in this, what we're going to talk about, is excluding those pests and diseases. I'm a real big fan of sterile technique. I understand you can't necessarily maintain a sterile technique all the time in a greenhouse or in a cultivation environment, but I really recommend pick up a tissue culture book. Plants from Test Tubes is my favorite, and just kind of have an understanding of sterile technique and try to implement it here and there when you can, and just have that mindset. See, I think I'm going to the next slide here. There we go. So definitely train your team to scout and monitor for pests. They need to scout and share their findings. If I walk through a greenhouse and I see an aphid and I don't say anything to someone, well, then they don't know about it. They can't plan their treatments, things like that. Communication is key. You got to empower them. Here on the right, you can see Corey walking through the greenhouse. He found this plant that had a branch that just had some necrotic foliage and kind of some funky growth patterns. And he said, "Hey, this isn't right. We're going to get it out of here." So he cut it and brought it up here. Knowing him, he probably sprayed it with ISO after that to try to keep it clean, reduce any transmission, and probably bagged it up and threw it in the green waste bin, that then left the greenhouse. So I definitely recommend that you find a good identification source, get familiar with it. There's some good books out there, the UC IPM website, other state extension services across the country. There's a lot of sources out there. Find one you like, find a bunch you like, get familiar with them, understand what you're going to be looking for. I really think all workers should have a high quality loupe or magnifying glass. I really like the 30x ones that have like about a 1-inch reticle, or whatever your lens, I guess would be the term for that. Definitely have your workers record or report their sightings. I think as beneficials as well as pests, that's really important. So you know what's there. You have an understanding of how to treat for them. Definitely teach those workers to identify beneficial insects as well as the pests. So if you look at these pictures down here on the bottom, this center picture, or the center left picture, we'll call it, just some aphids. You can definitely see their little spiracles there. Not 100% sure what species they are. If you look at this one picture to the right of that, you see this real round aphid right there. That's actually an aphid that's been parasiticized, has probably wasp larvae living inside of it, killing it and eating it from the inside out. It's a beautiful thing in nature. I would hate for that to happen to me, but I'm very happy when it happens to aphids like that. And then this picture on the left here is a Dyna Trap. Essentially, it's a UV light, and then it has a fan underneath it. And then the fan blows everything down to this little catch jar right here that has a filter on it. It's not going to fix all your problems, but I think it's a really good way to understand what type of populations of insects you have around. Definitely, if you leave that up, you will catch a lot of moths at night. And so if you're outdoor growing, you're catching the moths, you're going to reduce the population of pests that are there to lay eggs and then have caterpillars trying to eat your nugs. So that's just something to think about. And you can, of course, use sticky cards to monitor and trap your pests. So this picture on the left here is a little bit of caterpillar damage in an outdoor garden. This picture on the center top here is a green lacewing egg on a cannabis leaf. So that is going to hatch and turn into a beneficial. This picture on the right here is what happens when you let your dad try to grow some weed and he doesn't spray anything and doesn't know what he's doing. It gets lots of aphids, thrips, everything that could go wrong went wrong in that picture. The picture on the bottom here is a mix of Astargus and Venerate hanging out in a greenhouse up in Humboldt County. Thank you to Brian at Six Rivers Development for that picture. It's an awesome photo. It's just showing the combination, how if you use Astargus and Venerate together, it's a good combo for preventing, the damage and then the resulting rot from that damage. So we're going to talk about economic injury levels here real quick. Essentially, the economic injury level is a IP term used in integrated pest management. So it's basically a threshold when you would decide to apply products or pesticide products, things like that. So typically, let's say you're growing broccoli, you might be okay to have a little bit of damage before that damage is more economically injurious than the cost of using a pesticide product or a control product. So in cannabis, essentially, it's such a high value crop. It's relatively small cultivation sizes. You really want to have no damage, ideally. So that turns it more into a preventative talk versus a reactionary talk. So just keep that in mind. Preventative pest control strategies are the best. I see someone in the chat asking when the webinar is over. It's supposed to be over at 1. We're going a little long. We'll send you a link to the quiz as well, so you'll be able to take that. Even if it's not right after, you can take it later in the day, just so you know. So the best way to keep damage under control is definitely with a preventative IPM strategy using both beneficial insects and crop protection products. Often a weekly spray and veg and then early flower will keep pest populations low enough. A strategy I really like is in your cloning period, in your vegetation period, and then into your early flower, use pest control products and some beneficials here and there. And then when you decide, okay, let's say day 30, I don't want to spray anything anymore just because that makes me feel good, even though it would technically be safe. You then use some beneficial insects and beneficial mites and kind of have them give you some protection through the rest of your cycle. I tell people to spray smart. When I first came back in the cannabis industry after being in college, I walked into a greenhouse and someone was washing out a dram sprayer, similar to this sprayer in the center here. And I looked at him and go, hey, what's the REI on that? And he looked at me and said, what's an REI? An REI is a restricted entry interval. It's something everyone involved in pesticides should know and understand. Just something I like to put out there. Train your employees properly. Always use personal protective equipment. A hollow cone nozzle use high quality spray equipment. A hollow cone nozzle is what this dram sprayer here has on it. It's an adjustable gun, so it can go hollow or it can turn into more of a jet. Get really good coverage with that. But hollow cones are best for penetrating dense canopies. Cannabis typically is a very dense canopy unless you're de-leafing a lot, and then that's a whole nother discussion. So pick a good spray pattern. So if you have a greenhouse that has rolling benches in it, these plants are very tight, you have a small space. I like to walk from the front of the greenhouse to the back of the greenhouse, spraying the underside of the plants, and then come back and spray the top side of the plants on my walk back to the front. And I kind of keep that pattern. I know I get really thorough coverage and cover, you don't want to miss any sprays, especially with biological products. Thorough coverage is really important to get that good efficacy. If you're an outdoor grower, let's say you planted your plants pretty close together, you can't get a tractor through there, you can't easily hand spray it all. There's a lot of new technology out there, like this drone sprayer here, that could be a really good option for some of those, medium, large size grows. And then use appropriate surfactants as necessary. So surfactants or adjuvants are products that either change the spray coverage, aid in the absorption of that product into the plant, making it more effective. Sometimes they help with good coverage. So in this picture right here, I took this at a grow in Sacramento, had Regalia, Venerate, and then Oro Boost. I think Oro Boost is an awesome product. It's not our product, but it plays really well with Venerate and Grandivo. Helps spread our products onto the plant, helps them get absorbed into that cellular layer where they can be taken up. And it smells really good. It's a citrus oil-based product. So just something to think about. So here on the left is Grandivo. It's a fine water dispersed granule. And then we have Venerate over here on the right, which is a liquid formulation. Our CG line, Matt touched on it. It's differentiated from the ag line in that it has more testing and it has additional testing, and it has looser label verbiage. The products themselves aren't the actual product isn't any different in the jug. I just like to put that out there. So Grandivo CG is a water dispersed granule containing 30% dead Chromobacterium subsugii. So basically we grow this out in a fermenter, and then at that end of that process, we heat kill it. So it's really the fermentation byproducts that are the active ingredients, the magic juice inside that powder. 1 to 3 pounds per acre is the use rate for big applications. That typically is like 2 to 5 tablespoons per gallon of water per 1,000 square feet. 1 tablespoon of Grandivo weighs about 7 grams last time I weighed it out. So I would just, it's a good conversion factor there if you need it. You can do 4 to 10 day application intervals. We typically recommend alternating between, let's say, Grandivo and Venerate. They have slightly different modes of action, but similar target pests. So you can really have good results with that strategy. You can also rotate in other products. It's best to target newly hatched caterpillar larvae and nymphs and adults of sucking pests. You have the best efficacy if you use it preventatively. It's not super curative. You'll notice populations slow. And let's see here. Let's go to the next one. So the mode of action, I guess there's just little facts about it. So it's combinations of multi-products in there, multiple compounds produced by the bacteria. Contains dead bacteria plus those other compounds. Reduction in fecundancy, reduced egg hatch, and death via ingestion. It's selective in that it targets pests because they have to eat the product for it to be effective, but it has broad spectrum protection in that you're going to have aphids, thrips, mites, Lepidoptera, pretty much all the big pests it's going to affect. It is easy on pollinators and beneficials because those products have to be eaten. Typically, you don't want to spray any insecticides when, like, let's say bees are out and about in like an orchard or something. But if they come in contact with the residue, they're not going to be negatively affected. That goes for most biocontrol organisms as well. So Grandivo will give you agitation and repellency of the pest. Once they've been feeding on the product, they A, don't like to feed on it, and B, will have stomach issues. Sorry, I'm in a hotel and the room service is knocking on my door trying to kick me out. Give me 1 second. All right, sorry, I'm back. Gut disruption and death to the insects reduces the egg laying, the fecundancy of the insect or the live birth, like with aphids, and then they stop feeding as well. It's a liquid formulation. So Venerate, we're switching over to Venerate now. It's a liquid formulation containing heat-killed Burkholderia rhinogensis and spent fermentation media, so similar technology to Grandivo in that regard. It targets sucking and chewing insects and mites. You're looking at 1 to 4 quarts per acre foliar rate. That's like 2 to 4 tablespoons per gallon, typically per 1,000 square feet. 7 to 10 day applications rotated with Grandivo is a great strategy, and it'll target the if you target the newly hatched larvae and nymphs, you get the best control, but it will also have some effect on the adults. So venerates mode of action here. It has ingestion toxins, so basically it'll disrupt their gut. It interferes with their feeding. They don't like to be like piercing, sucking insects won't have the same pattern that they usually do. As far as their feeding, it does have residual control. So as long as that product will last, let's say 10 days on the plant leaf surface. So it has some long, not just immediate effect, it, affects them as they're eating as well. And it's safe to beneficials because it has to be uptaken into the gut. So for some specific recommendations for sucking insects, so thrips, aphids, whiteflies, and mites, you'd start your applications early. If you have populations that are predominantly adult, then use Grandivo. If your populations are predominantly juvenile, use Venerate. I don't know how much I personally agree with that. I would definitely say rotate them in most situations. For control of Lepidoptera, apply Grandivo or Venerate at egg lay or initial egg hatch. You can definitely tank mix our products with BT products, and I've seen extremely good results where you have dead bugs like 24 hours later in the field when you tank mix with a BT. I think it's a really awesome additive effect there. And Matt, we're going to pass this back over to you.
All right, biological solutions. Here we go. So I'll be covering some of the research that we've done with our products. And I've been chatting a little bit on the through the webinar, through the Teams chat there. So hopefully I can answer some of those questions we have when going about viruses, which are always a pain. So yes, let's talk about the first slide, which is, thank you. The efficacy of our two products, Venerate CG and Grandivo CG, on the two-spotted spider mite. So this trial, if you look at this trial, it's not the combination of the two products together. It's just either or of those products in that treatment, and the replications of that treatment. Okay. So on the left-hand side, you're looking at two large bars, the gray and a blue bar. The gray bar means that 7 days after application, and then the blue bar is 14 days after. So you're going to see that. So the untreated control number of motile spider mites or moving spider mites of 10 leaves for the untreated control were 200. At 7 days. And if you look at for Grandivo, there's basically 0 active or living spider mites for either the 4 grams per liter or the 8 grams per liter. Basically, these are all the 30 milliliters per gallon rates, or something equivalent. And then Venerate CG versus the Venerate CG, we also saw almost no living spider mites after applications made on a 7-day schedule. So these are sprayed every 7 days with one of the products that you see. Copa soap is another product that was looked at here at this Canadian research trial on greenhouse grown cannabis in 2019. And so they see that these products are working really well if you're using them. They take a little bit sometimes, but they do work, right? So let me go to the next slide here. And there's a little bit of a lag. There we go. Sorry, there's a lag in my computer apparently. Great. Okay, here's another slide here. Controlling root Mealy Bug with a drench of Venerate or Grandivo CG. Now this is before we're looking at the label to see what kind of labels what kind of things we're going to go after, or we want to go after for getting for label. And so currently Venerate CG only has a drenched label. The Grandivo CG does not have a drenched label. But if you look here, controlling root mealybugs over time, we have about over 90% control, almost up to 100% control of root mealybugs using our products over 3 drench applications, 7 days apart. Now, this slide, first of all, this slide is by Stanton Gill. He mentioned it earlier, and this is on sedum flaps. So he's looking at these, and it looks like it's definitely giving us good protection or control of mealybugs. So when we're looking at the control of 2 spotted spider mites on cannabis, what we're looking at here is another trial that was done in 2018 at the Greenhouse Cannabis in Canada, a research facility there. And this is a number of modal or living, actually moving, living 2 spotted spider mites that are on this cannabis plant over a period of time, applications. And what we noticed is that both Venerate and Grandivo did a great job of really controlling spider mites. Again. All right. So I think what we're going to do now is have maybe some question and answers. Angela, I'll let you take it from here. Things are resources, contacts, question and answers. Aha.
Yeah.
Here we go. Well,
so I'm gonna.
I can.
Hey, I'm gonna jump in and answer this question.
Materials.
I mean, while that's up, I'm gonna answer this question. Maria, I think, or Maria, I don't know how to pronounce that. She asked, when would you use Stargas versus Regalia for bud rot resulting from caterpillar damage? So I would be using Regalia preventatively to really have that plant's immune system at its best, I guess would be the right answer. Reduce the amount of damage you're going to see. Once you have caterpillar damage, though, it's almost too late. You really should be using a product that's going to prevent the caterpillars from damaging the, like, let's say Venerate or BT spray. If you mix in the Stargas with the BT or the Venerate spray, what you're doing is you're preventing the damage. And if we'll say reducing the damage. So let's say you still have 1% of caterpillar damage. That Stargas should limit the amount of botrytis that is going to be forming in that nice tissue that's been damaged and now is dead and in that humid cola environment. And ideally, you would also pick off any dead colas that you see to reduce the spread and reduce the level of inoculum present. I think that answered that question. Sorry for jumping in there, Matt. And Venerate does have some soil limits, some soil application limits in California specifically. Our DPR is a little bit stricter than the rest of the country. So
that's right.
Just,
and for everyone who's still on, yeah, sorry to jump in, but for everyone who's still online, no matter where you're coming from, every unfortunately, most states are all different on what they're allowing for use on cannabis. So just make sure that all of the products that we're talking about here in our webinar, just make sure that they are available for you in your locality.
Awesome. I just wanted to share really quickly some resources for you guys. If you're happening to watch this on your desktop, you can take your mobile device, open the camera app, and just hover over the QR code. That will automatically open up. It's going to usually actually bring a drop down, and you want to click that drop down. That'll take you onto the website where this cannabis pest guide is. So that is often a really good resource. The next set of resources that we have on our website are our Hemp IPM guide. So that's focused on outdoor hemp. So the QR code is on the left. Again, just open your camera app, put it over the QR code. Drop down will pop up. So you want to click on that drop down, and then you'll be directed to the web page where you can download that. And then on the right is our new Cannabis IPM guide. So really a step-by-step process to walk you through what we believe are the best practices for managing your crop from transplant to flowering stage. And just a quick review, Nic Steeneken, who you heard from on this webinar, is over on the left in Northern California, Nevada. And we have the rest of our teammates throughout the country. Matt Breck is a national account manager, so he's available to anyone. And then we also have contact information here. So these are good to just take a quick screenshot or photo of. They're all on our website as well. If you go to marronebio.com, look in the upper right-hand corner for sales team, click on that, and you'll see everybody's contact information. We have technical support throughout the country, and that concludes our webinar. So Brenda, I'm going to ask you to go ahead and put the link to the quiz in the chat. For anyone that got off the webinar, obviously you're not hearing this now, but you might hear it in the recording. Just go ahead and take the quiz. I will go ahead and email all our registrants as well, so in case somebody hopped off. Are there any final questions that our team can help answer? Otherwise, let us know if this information was helpful. And again, we will try to do a follow-up, you know, it might just be a live Q&A or it'd be a really brief webinar. Once Dr. Punja is able to join us again, we're so sorry he was not able to join us. It was very last minute, unexpected occurrence. And of course, if you have any questions or concerns or suggestions for future webinars, we're always happy to hear that. Great. My colleague Brenda has shared her email address. She's also shared the link to the quiz. You guys, while you take the quiz, I wish SurveyMonkey was better. For some reason, they don't provide a confirmation after you submit the quiz. So it'll just say all set or something like that. But most often we always get the results. We haven't had anybody take the quiz and it's like magically not, you know, we haven't received the results. So just know that sometimes the final experience in that after you submit the quiz questions is not always very clear. The quiz link is just above in the chat. So just look in the chat. It's the SurveyMonkey.
And I will note for anyone that needs DPR credit, I believe, Brenda, correct me if I'm wrong, that we submit it so it should show up on your hours report through CAPCA. I'm 99% sure this is correct.
I'm not sure about CAPCA, but we do have CCA credits.
Okay. So link didn't load. It's loading for me. Perhaps you have to hit the control button and then click.
Can we email it out to everyone that registered for the webinar here?
Yep, we will.
Afterwards as well, so they have it. Okay.
So yes, everyone, the recording, a copy of the presentation in PDF form, as well as a link to the quiz will be coming out. It'll either be later today or more likely tomorrow. So there will be a delay. It takes a while for the downloads. So a couple of people are saying the link isn't loading. So I'm not having a problem, but I'm doing this off of my phone. Is there sometimes if you click on the link and it doesn't load, you have to press your control button and then click? So we've got a lot of people. There we go. Thanks. Thanks, friends. Copy and paste the link into your browser. Technology is just not on our side today. So I just have to laugh. We did a webinar yesterday and we've had no problems for some time now with webinars, but this is what happens every once in a while. Technology just doesn't want to cooperate. Awesome.
Hey, we might have had some technical difficulties, but I think this was, as far as information wise, one of the most well-rounded webinars we've ever done. So I'm happy with it.
Good. I'm glad you are, Nick.
Thank you to everyone that was a part of it. We appreciate it.
It was a team effort for sure. Okay. Yes, you can take the quiz later, you guys. You have several days to take the quiz. Just don't come back to us in December wanting to take the quiz. But by all means, if you need a little time or even a couple of days, we usually pull the results. That's why we wait to issue certificates of attendance. So typically we try to issue the certificates of attendance 1 week after the webinar. So please just make sure you complete your quiz by like next Wednesday, okay? Awesome. If you guys are all set, there's no reason to hang on the webinar. I just am going to hang on along with Brenda to make sure nobody has any other questions or problems with the link. Otherwise, we look forward to keeping in touch with you guys via electronic communication. You can expect a recording of the webinar, PDF of the presentation, and the link in your email as a follow-up. Go ahead, Matt.
Yeah, I'm just staying online. If anyone had some final questions, to make sure everyone gets their points and credits, quizzes, and such. There's some questions that came in. Some of these questions that are coming in, guys, on the chat. Love questions. I'll answer them all day long if I have to. Is that there's some of them are a little longer than the chat, so give me a call or give Nick a call. We can answer them. It's going to take a little too long to do like little text message. Some of these questions are really in depth, but that's what we're here for.
Hey, Mohammed, where are you located at? I would reach out to whichever rep is in your area for a possible Stargas sample. We're really big fans at Marrone's of doing grower demos and really putting our product in your hands and showing you how to use it properly. So that's most likely possible as long as the product is registered in your area. Then you'd be reaching out to Matt if you're in Ontario.
Yeah, you just give me a call. There's a question came up about from Brent said, or he said something. I'm pretty sure Mr. Gill said he found crypto feeding on root aphids. Anyone hear that before? Personally, I had not. And you talk about cryptolaemus montroseri is what I believe is what Brant is referring to. So that is a first for me. That is one of those things I'm going to be talking with Stanton here soon. But anyone else have any comments on that maybe in the chat? Or Nick, have you seen that?
No, but I know they're pretty voracious predators is my understanding, and it wouldn't surprise me very much that they would do that.
Okay, yeah. How many people do we have online? I don't know. We have
We're 34 right now. Yeah.
34.
Yeah.
Got it. Okay.
Typically, we have a handful of people that just linger on because they're busy doing other things or whatnot.
Yeah, no, for sure. I just want to make sure that we're getting everyone's questions, while we're here, while we're online. Thank you so much for everyone who said thanks and support. Really great. We love to put these webinars on. We learn things just as much as you guys do, because a lot of this information and the research is really coming as we go. All of this is new, market, new research. As we had to have federal legal legislation go through in order to do full, sort of university research, the funding. But there are some that are able to do through certain special grants in their states that have legalized it, I know. And most of it is done on hemp or some kind of hemp plant, which is basically the same if you want to think about it. And some of those cultivars, for that matter. Let's see, go through this here. Yeah, the QR code. How's that going? I know we try to make sure things are really easy to use, and that QR code I hear is pretty good.
And all those stuff that we link to is available on our website as well. If you go to marronebio.com, you can always find it from there as well.
So I might as well just go ahead and, if you want to call me, or Nick. But one of the questions comes up about, like when you're timing for bud rot, and let's say corn worm, or those, Lepidoptera, a worm or cutworm or something, some kind of worm species, to come in and eat and lay those eggs, or mollusks lay those eggs. The larvae hatch and then go right in and start damaging the colas. The timing is basically when those moths are laying those eggs. You have to make sure you know when those eggs are hatching. So the eggs apparently turn dark color, maybe like a blackish, maybe like a dark, like a really dark brownish black, and they start basically they erupt and you know, the larvae come out and they go forth. And you know, that's the timing right there when that that first, you know, the first egg hatch happens. That's a perfect timing to go out with your bioinsecticides, because that is when these insects are most vulnerable. Okay, and so the practice is basically is that because moths will tend to, you know, lay at certain times. Sometimes they'll lay by the moonlight, a certain time of, you know, term of the month, or they'll delay just because it's just sort of that perfect environmental conditions, humidity and temperatures, right? And or they have just a great host, like you know, let's say they were in a cornfield and they went over to next door to the hemp field that's now blooming and smelling great, and that's what happens, you know. But when that flower starts, you know, producing that cola outdoors or in that greenhouse, and you know, you have moths that are flying around, they will land on those colas, lay their eggs, and then those larvae go in. If you spray Venerate plus a BT product, you can pretty much control most of your issues right there with those caterpillars. And what we also say is, you know, that timing is exactly when the timing is for Astargus as well. So and when that is, you know, it's going to obviously determine when you're growing outside or in that greenhouse. And if you don't have covering, if you're growing in a greenhouse, definitely make sure you have got some kind of screening to keep out moths and other insects that will really reduce how much issues you've got with Lepidoptera and Botrytis Bud Rot. But the timing, you know, for me, when I'm looking out, you know, for hemp or let's say cannabis production outdoors or in a greenhouse that has like, you know, rolled up sides, or a hoop house, that timing is basically like week 3, what we call indoor. So like you're just having some buds coming in, and you'll have some smell there of the flower. That typically like week 3 and 4 is this kind of the set was where I start to see some of these applications go out for the for not only the caterpillars but also the Botrytis Bud Rot, because if you're going to put it out, you're going to put out every sort of 7 days or 10 days from that initial point to maybe, let's say, week 5 or 6 or 7, depending how long you're going to take that in your greenhouse or outdoors. And you may only need like 2 or 3 applications, but the Astargus will get inside that cola as it's forming and sort of coalescing and going up and basically filling in. And that's when that Astargus can basically colonize those buds and prevent any other colonization from happening to from other diseases, specifically Botrytis Bud Rot and Fusarium.
Hey, I'm just going to jump in and say that I'm going to have to take off right now. And I really appreciate everyone for you being here and hope you guys all have a great rest of your day. And take care.
That's
awesome. Thanks, Nick.
Signing off.
I'm going to go ahead and sign off as well. We'll stop the recording. If anybody's lingering on and happens to have some questions, don't hesitate to contact us through the emails that were sent to you for this webinar. Thanks again for joining us and have a great day.